Hi there. You may be wondering why I'm running around this park dressed like your mum's Zumba teacher, but I'm doing it in the name of science. And running is a very complex science. The heart is this great, big, muscular pump that only has ever had one aim and one dream in life: to pump blood around the body while maintaining pressure. It pumps glucose, nutrients, oxygen and hormones around the body to respiring tissues in order for us to survive. During exercise, your heart has to maintain blood pressure to ensure that your muscles in your legs, your arms and your chest receive the appropriate amount of glucose and oxygen required to enable them to contract. But not only this, the brain has to receive that appropriate amount of glucose and oxygen via the pumping action of the heart to ensure we have the motivation to continue after 30 seconds and not snack on the flapjack in the gym bag.
In order to understand how the heart's pumping activity maintains blood pressure, you need to be familiar with the four chambers of the heart: the right and left atria and the right and left ventricles. The right atrium receives deoxygenated blood from both the superior and inferior vena cava, whereas the left atrium receives oxygenated blood from the pulmonary vein. On the other hand, our right ventricle receives deoxygenated blood from the right atrium and pumps blood out of the heart to the lungs via the pulmonary artery. Finally, the left ventricle receives oxygenated blood from the left atrium and contracts the pump blood out of the heart through the aorta to respiring tissues around the body. Looking specifically at the ventricles, the left ventricular wall has a far greater muscle mass compared to the right ventricle, so that it can generate greater force. The left ventricle is pumping blood through the entire body, compared to just the lungs. Therefore, needs to generate greater power and pump blood at a higher pressure. So, what regulates this one-way flow of blood into the atria, through to the ventricles and out of the arteries? Well, between the atria and the ventricles are the atrioventricular valves, which function to ensure when the atria contracts, blood can freely flow into the ventricles in a unidirectional movement. Likewise, the semilunar valves function to ensure a one-way direction of blood flow from the ventricles to the arteries during ventricular contraction. All these dynamic structures are working in coordination to ensure that pressure is generated to pump blood to the lungs and around the body. The cardiac cycle is a series of pressure and volume changes that take place within the heart. This can be divided into three distinct steps. 1. Atrial systole, contraction of the atria. 2. Ventricular systole, contraction of the ventricles. And 3. Both atrial and ventricular diastole, relaxation. Atrial systole. During atrial systole, the atria simultaneously contract, increasing the pressure inside these chambers. Contraction of the atria reduces the space inside the atria, which increases the pressure whilst the volume of the blood stays the same. This high pressure exerted in the atria will force the atrioventricular valves to open, so blood flows into the ventricles down the pressure gradient to an area of lower pressure. Ventricular systole. When our ventricles have filled with blood from the atria, the pressure therefore increases within the ventricles, causing the atrioventricular valves to close, which prevents backflow into the atria, like a cardiac bouncer preventing ventricular blood re-entering the atria which they have just been kicked out from. Much like the atria, the ventricles contract and the space within the ventricles decreases. Increased pressure is now exerted on the blood. The pressure in the ventricles is now much higher than in the aorta and pulmonary artery, which forces the semilunar valves open and blood to be pumped out of the heart at a great pressure. Now the pressure within the arteries is greater than in the ventricles, causing the semilunar valves to close, preventing backflow into the ventricles. Atrial and ventricular diastole. Diastole, the relaxation of the atria and the ventricles. Following the contraction of the atria and the ventricles, the pressure drops. This is due to the volume of blood within the chambers decreasing and the muscle walls relaxing. As a result of diastole, blood can re-enter the atria from the vena cava and the pulmonary vein, resulting in the initial increase of atrial pressure seen at the beginning of the cardiac cycle.
The cardiac cycle can't happen alone. The contraction of the atria and the ventricles is controlled by electrical impulses. Think about it like the perfect pairing: yin and yang, peanut butter and jam, cheese and ham. Without electrical impulses, cardiac muscle would simply be unable to contract, disrupting the cardiac cycle. Cardiac muscle is myogenic, simply meaning the heart can generate its own electrical activity without any nervous stimulation. These intrinsic electrical impulses mediate coordinated atrial and ventricular contraction. In the right atria, there are a collection of nodal cells which make up the sinoatrial node. These cells initiate a wave of excitation through the atria which spread out over the muscle walls of both atria, initiating atrial systole. Therefore, it is unsurprising that these sinoatrial nodal cells are often referred to as pacemaker cells, which initiate the rhythmic electrical impulses across the atria that regulate heart rate. In a healthy individual, resting heart rate is usually maintained within 60 to 80 beats per minute. This spread of electrical excitation is prevented from passing over the ventricular walls by the atrioventricular nodal cells. This allows for the atria to complete its contraction and emptying of blood into the ventricles. The atrioventricular node then relays the signals to the bundle of His, which are fast-conducting muscle fibers located within the connective tissue between the ventricles. The bundle of His is divided into the left and right bundle branches, which then direct the wave of excitation to the apex of the ventricles. Here, the wave spreads out through the Purkinje fibers of the ventricles, causing them to contract and push the blood upwards towards the arteries. This coupling between electrical excitation and systole maintains the regular and effective cardiac cycle. Fortunately for you guys, we can monitor and record the heart's myogenic electrical activity during the cardiac cycle. So, stay tuned for some casualty-style beating and some groovy waves.
We have all come across the infamous ECG flatline sound from our favourite casualty TV shows and those dramatic peaks which indicate whether you are dead or alive. The electrocardiogram, or ECG, is a simple test which can be used to monitor the electrical activity of the heart and therefore diagnose and monitor cardiac conditions. Each individual wave has distinct sections: the P wave, the QRS complex, and the T wave. The first wave is referred to as the P wave. This wave represents depolarization of the atrial muscle cells, or in other words, the electrical excitation of the atria, resulting in the contraction of cardiomyocytes. The second distinctive section of the wave is the QRS complex, which represents depolarization of the ventricular muscle cells. And lastly, the T wave, which represents the rapid repolarization of the ventricles. This underpins the relaxation and consequent refilling of blood during the cardiac cycle.
Thank you, Seren. So now let's take a look at how we set up an electrocardiogram to monitor the electrical activity of the heart at rest and during exercise. So here we have 3 ECG electrodes that act as small sensor pads. These will be applied onto a skin to enable the detection of electrical activity of the heart from the surface of the body. This activity is then transmitted to the monitor, amplified and displayed on the ECG trace. So let's start with the earth electrode. The earth electrode is placed on the right leg. Secondly, we have the positive electrode, which is the black electrode, on the left leg, down towards the ankle. And finally, we have the negative electrode, which in this case is the white electrode, and that's placed on the right wrist. And now we have a basic 3-lead, lead 2 ECG setup. When Seren is at rest, we can see these regular electrical wave formations like the ECG wave we looked at before. The ECG shows clear P waves, QRS complexes and T waves. The number of QRS complexes per minute represents the heart rate. So let's see what happens when Seren hops on the bike. As you can see, the number of ECG waveforms dramatically increases, and the distance between the QRS complexes dramatically decreases. This is because Seren's leg muscles are now contracting to keep the pedals moving and therefore using oxygen and glucose at a higher rate to maintain these contractions. The heart rate and force increase to supply the demand for oxygenated blood by Seren's legs is regulated by an increase in the frequency of waves of excitation generated by the sinoatrial node, the nervous system stimulation, and circulating adrenaline. That's it from us today, and we've covered the
Heart structure and function,
cardiac cycle,
the electrical activity,
and the ECG.
bye-bye from
See you later.
the Brainiacs.
The Brainiacs.